Hello, my name is Wei Kheng Teh, and I'm speaking from MBO EBI. And today I'm going to be talking about towards standards for harmonization and reproducibility of single-cell data. Particularly, I'm going to be talking about the challenges towards interoperability in the single-cell omics space. The major challenge towards interoperability in single-cell omics is that of heterogeneity. We have heterogeneity of different metadata standards, for the community has yet to converge on one. For example, FANG, MINSC, HCA-DCP, and also the granularity of those metadata standards. Some standards may go to the level of the cell and contain cell type information, but other metadata standards may only stop at the file level and only contain metadata on the file. This presents another challenge to interoperability. There's also heterogeneity in file formats. You have many different file formats, Loom, Surat, ScanPy, and these are also not easily integratable with each other, and coding languages. Different packages can only work in R, some only work in Python, and there's a potential to interoperate between both of them, but there is still a significant barrier there. And finally, there's a heterogeneity in terms of where you deposit and find your data. You can deposit spatial transcriptomics data in the bioimage archive, but also expression data in the single-cell expression atlas, or specific deposition and archives and data coordination portals for the apps such as the Allen Brain Map or the single-cell portal or DISCO. And this results in a lot of single-cell omics data being very disparate, being found in lots and lots of small repositories and smaller silos spread out across, and not having a comprehensive place where you can find all single-cell omics data linked to each other with metadata and data. Now, this issue of heterogeneity is not unique to the single-cell community. However, it is particularly pertinent to the single-cell community because the community has converged on this final analysis file, which has embedded metadata within the file. What does this mean? This means that researchers and scientists are taking a single analysis file, uploading it to a visualization portal, and would like to plot out their UMaps and see the results of their analysis. And they would also then like to be able to find sample level metadata, library level metadata, and cell level metadata embedded in that single file, not separate from one another. So you'd be able to look at your analysis and also see what characteristics, what age, what sex, what gender, what other metadata can be appended to that specific analysis. This means that every variable that changes results in this almost exponential explosion of potential final analysis files. You could have all of the similar metadata standards in the files, but they'd be in different file formats, and that would not be interoperable, or vice versa. You could have all of them in the same file format, but in different metadata standards, and that would also not be interoperable. Those are the two issues that I really want to speak about today, and what I see as the largest obstacles to single-cell omics interoperability. So there's an issue with file formats in terms of interoperability. For example, we have Surat files, ScanPy files, and Loom files, which are not interoperable with each other. However, there is significant tooling which exists, such as ScanPy, in order to convert these file formats to the same file format. Great, they're now interoperable. However, these file formats also store metadata in different metadata standards. You could have 3 Surat files, each using a different metadata standard. So they would store metadata in different headers, they would maybe use different ontologies, they would have different levels of granularity, and that would mean that they are also not interoperable. You can't easily and accessibly integrate those files together. And we see this as a particular issue because of the lack of awareness and the lack of tooling surrounding issue with metadata standards. There's lots of tools and lots of people working on converting file formats, but there's a significant lack of attention and tooling for converting between different metadata standards. And we think that this isn't a problem which is difficult to solve. This is, in fact, a problem which could be solved fairly easily. And I'm going to be talking briefly about a poster which I've created, which specifically points towards this issue. This is a poster which we presented at the ELIXIR All Hands meeting in Uppsala, Sweden in 2024, which specifically talks through this problem of harmonizing single-cell data in a crowded and disordered data ecosystem. So there are tools such as SCEZ or SURATDisk, which convert the process count matrix to the same file format, but you get different unaligned metadata, and this can lead to extensive data cleaning or exclusion of available but unaligned metadata from the analysis. These still are not interoperable. Our suggestion was a single-cell omics Rosetta Stone. This would function as an adapter that aligns the metadata based on expected community standards and outputs the aligned metadata as a single file in a user-defined metadata standard and unit. So you would be able to upload or select a number of different files of different file formats and metadata standards and be able to receive those files in the format that you wish and the metadata standard that you wish. And this interoperability of data standards, providing this harmonized data, means that we don't have to spend an extensive amount of time and money data cleaning or leaving out metadata from our analysis. This would obviously start with a single use case of polling the community and discovering which standards are most useful to be interoperable and be able to adapt between. And we would love to have more of the community speak to us, especially at the ELIXIR single-cell omics community calls, which happen every month. It would be great to have your input, your feedback, and to work together on fixing this problem. Thank you.